Bun găsit la emisiunea Știință și Cunoaștere. Azi ne reîntâlnim cu Bruce Lipton pentru un al patrulea set de videoclipuri care răspândesc mesajul său de optimism și sănătate. În perioada ultimilor 2 ani nu am mai avut ocazia de a difuza interviuri, deoarece profesorul Bruce Lipton are un orar foarte strâns. Dar domnia sa mi-a oferit drepturile de utilizare și difuzare a oricăror din materialele produse sub numele său, adică videoclipuri, DVD-uri, cărți și articole. Din când în când, Bruce Lipton oferă abonaților săi de pe contul de YouTube scurte prezentări ale unor idei esențiale. Le-am colecționat pe întreaga durată a pandemiei cu scopul includerii lor în câteva episoade Știință și Cunoaștere. Am realizat integral traducerile, subtitrările și sincronizările, așa că nu-mi rămâne altceva de spus decât vizionare plăcută cu o nouă înțelegere a biologiei în prezentarea condensată și ilustrată de către profesorul dr. Bruce Lipton. Doresc însă să vă reamintesc și de această dată faptul că în toate aceste videoclipuri produse de Bruce Lipton există foarte multe metafore. Asta înseamnă că dumneavoastră aveți nevoie să înțelegeți sensul metaforic al ideilor și frazelor sale. Acolo unde a fost posibil am adăugat ghilimele sau anumite note între paranteze. Dar în multe alte situații nu a fost posibil și de aceea rugămintea mea este de a acorda multă îngăduință și atenție la vizionarea acestor materiale. A doua problemă este legată de viteza foarte mare de vorbit pe care o are profesorul Lipton și care este un adevărat maraton. În multe locuri a fost nevoie să fac adaptări pentru a scurta numărul de cuvinte afișate simultan pe ecran, dar chiar și așa s-ar putea să nu fie de ajuns. Din acest motiv, vă rog să revizionați aceste emisiuni ori de câte ori se vor putea redifuza la TVR Cluj pentru a pătrunde esența tuturor ideilor sale, având în vedere faptul că unele lucruri au scăpat atenției dumneavoastră. În primul material, Bruce Lipton ne prezintă influența epigenetică asupra comunității de celule care alcătuiesc corpul, dar care este dublată și de un răspuns al minții conștiente asupra evenimentelor de viață. Asta înseamnă că avem influența mediului ca factor indirect și la aceasta se adaugă influența minții ca factor direct și esențial. Dar mintea noastră este structurată în 2 compartimente, conștientul și subconștientul, care lucrează cu programe. Este foarte interesant de observat analogia pe care Bruce Lipton o face referitor la o intenție conștientă, dar care nu are și un program subconștient implementat. Iar în acest caz, subconștientul face o autocorecție asemenea dicționarelor digitale din telefoanele mobile și tablete.
Hi, dear friends, cultural creatives and seekers everywhere. Bruce here with a very important distinction to be made, and that is about the nature of epigenetics and the environment. What do I mean by this? Well, I understood the nature of epigenetics back in the late 60s, early 70s, about how the environment of the cells were controlling the genetics of that cell. And it took up until the 90s before conventional science accepted this and actually gave us a meaning behind the new science of epigenetics. Environment influences cells. And I go, this is absolutely true. But then how come in biology belief I talk about our mind and our perception controlling our genetics? If a cell is out in the environment, consider an amoeba moving through the environment. Indeed, all environmental influences are now going to affect the behavior and activity of that cell. But then I say, well, wait, do those cells actually see the real environment in which we're in? I go, no, the cells in your body are covered up by the skin, disconnected from the environment. But in order to survive, these cells have to adjust their genetics and their behavior to adapt and maintain their survival in a changing environment. So the question is, then how do the cells inside your body actually know what's going on in the real world because they're not connected to it? I go, this is the role of the nervous system. The nervous system's job is to read environmental signals and then relay those signals into the cell. So as illustrated right here, we are getting signals from 2 different environments in our body. Signals from within our body as well as signals from without our body, and the function of the nervous system is to read both of these environments. The relevance is the nervous system reads the information in the environment and then translates it into signals that are sent to the 50 trillion cells inside your body to alert them to what is going on in that world. So, in fact, that is a true statement. The environment is controlling the behavior of your cells. But the mind does an interpretation of those signals based on our life experiences. Now, for a lot of people, this seems difficult, but let's put it in a different context because we can talk about computers. There's a very interesting fact about our brain as a computer because it has an autocorrect mechanism built into the subconscious. Meaning what? Well, I can have my conscious mind like typing on the computer about what's going on in the world. But if I type something, that signal can be translated and sent to the cells. But if I type something that the subconscious has no recognition of, it will do like an autocorrect and change what I wrote to fit something that it thinks that I wrote as a subconscious. So I put in love and subconscious changes that to live. That's a whole different meaning at this point because of misinterpretation. So the point is, your wishes and desires of your conscious mind are filtered by the programs in your subconscious mind. And if you have a wish or desire that doesn't conform to a program that already exists, then the subconscious mind's job is to retranslate that into something that makes sense for the subconscious belief, not for the conscious. So we may have wonderful wishes and desires, and if it doesn't conform to the programs that we receive, the subconscious will change those into something it understands. To make sense out of what I just said, consider using your cell phone. That you write something in your cell phone such as, let's put the word epigenetics in the cell phone. And I go, when I type epigenetics in, the database in the phone says, I don't know that word at all. And then what does it do? It throws up different options that could be closely related to epigenetics. And so the significance is, if I put something into my system that is not supported by the program, then the programming system will change whatever I did to accommodate what it believes is right. Now, the issue about this is, well, if I want to have epigenetics in my phone, then what I really have to do is open up my dictionary and add the word epigenetics. And now after that, anytime I write epigenetics, the phone mechanism will understand what I'm talking about. Well, going into the database and changing the program is the function of what energy psychology is all about. It says if your behavior is not conforming to your wishes and desires, then it will be necessary to go into your program and install new behaviors that you want to manifest. So now the brain will have a reflection or a resource of what you're talking about. So I really want to emphasize the nature of biology belief as different than the fact that just environment controls your genes. And I'll give you a very simple example of what I mean by that, by the fact that I can have two people in the very exact same environment, but each of these individuals has a totally different reaction to the environment. So if it was just environment that controlled your genes and your behavior, then both individuals should have exact same behavior. But the reality is no. The two people have different responses to the environment. I go, why? Same environment, but their minds have different interpretations. And it's the interpretation of the mind about that environment that is sent to the cells. So the important part is this. Yes, environment controls epigenetics. There's an external environment, but an internal environment. And yet recognize in humans, the internal environment is not a direct readout from the external world. It's an interpretation of that external world. And that's why each of us has a different response to the world in which we live. And that is why the biology of belief is an extension of the foundation that environment controls epigenetics.
Următorul material pregătit de Bruce Lipton se referă la modul în care noi ne folosim de resursele naturale pentru a face o comparație cu modul în care ne folosim propriile resurse biologice. Din această comparație rezultă faptul că asemenea folosirii greșite și abuzive a resurselor naturale care ne amenință cu extincția, tot așa folosirea greșită și abuzivă a resurselor noastre biologice ne amenință cu boli și suferințe. Bruce Lipton spune următoarele: noi suntem natura și nu controlorii naturii. Iar prin comparație cu structura biologică, Bruce Lipton ne spune, Armonia în comunitatea celulară înseamnă sănătate, iar disarmonia în comunitatea celulară înseamnă boală.
You know, there's an interesting story in the Bible. It's called Genesis. And in that story of Genesis, God creates the planet, God creates nature. And as an afterthought, more or less, God created people and put them into that mix. The unfortunate part of that story that's been carried around for the last couple of thousand years makes a situation that suggests that we and nature are separate from one another, that we came on our own separate from the planet. Well, this has been a big problem. You know why? Because we are actually created by nature. And the significance is, as the fate of nature goes, so does the fate of human civilization. And now comes the biggest problem is that humans have been destroying nature and precipitating what is called the sixth mass extinction of life. People just haven't caught on to the important part. If nature goes down, so does humanity go down, because we are nature. So maybe it's the wake-up call, a wake-up call that actually is a call from a long time ago. 10,000 years ago, the indigenous people on this planet already knew that nature was a garden, and they were here as gardeners to help create the beauty of that garden. Well, let's wake up to that original belief, because that's what's actually controlling our world. We have to stop using the earth and start working with the earth. That's how our evolution will take us into the future. An interesting concept of humans is that we are the smart, intelligent creatures of the planet, and the rest has less or even no intelligence to it, just reflexive behavior at some point. But this is not true. As a matter of fact, here's an important point. The human body is made out of cells. The cells are the intelligent part of the human body. We are just a collective expression of cellular intelligence. Well, nature is imbued with all this intelligence. And the fact is this, when we start to take the stance that we have dominion over nature is when our civilization started to fall from grace. Significance why is we are nature. We are not the controllers of nature. We're the ones that are supposed to learn how to live in harmony with a garden. Simple fact, a garden is not a battleground. But look what we did. We've turned it into a battleground. A garden is the height of cooperation. And this is what nature is asking humans to do at this moment. Wake up and start learning to live in harmony and cooperation with nature. Well, guess what, folks? You've been looking at yourselves as a single entity, a single organism, but when you look in the mirror and you see that single organism, let me adjust the understanding, and that is this. A human is actually a community of 50 trillion human cells. In fact, there's even more cells. We call it the microbiome. You're covered with 10 times as many microbiotic cells, such as bacteria and fungus, and these are part of our life. Guess what? You can't live without them. So today's definition of a human is not just the human cells, but all the microbiota that live with us, because without them, we will not survive. In human health, we're not talking about the individual health, we're talking about the health of a community of those 50 trillion cells. And then I go, well, they are miniature people, these cells. Why? They have all the same functions that you have: respiration, digestion, excretion, nervous system, even immune system. And so what's really important is your health is not based on one cell, it's based on 50 trillion cells living in community. And the significance of all that is then who's organizing this community? And it turns out the brain is organizing this community, and the significance is very clear. The brain doesn't tell the digestive system how to do its job, or the heart how to do its job. The brain is coordination. And if we screw up this coordination, then the community begins to fall apart. So it comes down to a simple fact. Harmony in the community is health. Disharmony in the community is disease. And all of a sudden it says if we want to heal ourselves, we don't have to worry about the genetics of the individual cells. It's actually the consciousness, the government by which 50 trillion cells are living their lives. How are your thoughts these days? Are they positive? Are they negative? Why? Because your thoughts are a political voice for 50 trillion citizens under your skin. And when your thoughts are in harmony, so's that community. So let's look at health in a different way. A political situation versus a molecular genetic situation. It's the politics of health that we understand. You know, when I was a professor in a medical school, what was I teaching? I was teaching about this organ system and this one, the respiration system, the digestive system, the musculoskeletal system. We looked at all of these as little independent systems to study. But guess what? Over time, we learned something very important. You cannot study an individual organ alone. It's a process of community where all the organs are integrated together. So there's a whole different approach today. It's called systems biology. I'm not just studying the heart. I'm studying the heart in relation to the brain, the heart in relation to the gut, the heart in relation to our behavior. So all of a sudden we start to see that every organ system is connected to every other organ system and they work as a system. And so the beautiful new view is not to study how a single organ works, because it's connected to everything else. And as a result, if we understand the interconnectivity of the organs, then we can understand the whole process that comes out of a human. So we're going from reductionism, studying the pieces, into holism, studying how all the pieces come together and create a whole. And that knowledge is empowering. And if you understand it, you can have more power in creating your life.
În continuare, Bruce Lipton dorește să ne explice care a fost drumul său în cercetare, cum a ajuns la epigenetică, cine l-a îndrumat și care au fost lucrurile ce i-au schimbat viața. Materialul următor ne dezvăluie un lucru foarte important și anume, citez, înainte de a da vina pe celule pentru patologie, să recunoaștem un fapt simplu, și anume, doar 1% sau de fapt mai puțin de 1% din boli este legat de gene, peste 90% din boli sunt legate de modul în care răspundem la condițiile de mediu și evenimentele de viață.
"55 years ago, I ended up at the graduate school at the University of Virginia in the biology department. And I was working for one of the most interesting and profound scientists of the day, Erwin Koenigsberg. He was one of the fathers of the cloning technique of stem cells. So while 55 years ago, hardly anybody in the world even knew what a stem cell was, I had the greatest opportunity of not just knowing stem cells, but actually growing them and cloning them in tissue culture. When we talk about stem cells, there's several different varieties of stem cell. A basic stem cell is called a totipotent stem cell. That means total potential to grow any kind of cell in the human body, be it skin, muscle, bone, brain. These are basic stem cells that can replace anything. But the research I was working on was on a more restricted developmental stem cell, and it's called a myoblast. This is a cell that you can identify early in the embryo that will become the precursor of the muscle in the body of that embryo. So my research was to take out myoblasts and clone them and grow them in culture. And you say, but why? And the answer is very profoundly important, because basically we needed to have an insight in how does muscle form? What are the mechanics that control the development of muscle? More specifically, actually, it was the funding that I was getting from the Muscular Dystrophy Association to do this research for a very important reason. In muscular dystrophy, there's a breakdown of the muscle, and there's a problem with the regeneration of that muscle. So by working with the stem cells that generate muscle, we were going to get some insight into the mechanics of how to repair muscular dystrophy problems. My major advisor, Erwin Koenigsberg, gave me some advice at that time that I didn't realize was so powerfully influential on the rest of my life. I didn't even understand, I didn't even see it as it came down. And I said, what was that advice? And Irv told me, when I plate the cells out into my culture dish, that if the cells don't look right and they form these irregular looking cells, he said, before you blame the cells for the problem, the most likely source of the problem are the conditions of the environment, the culture medium in which I grow the cells. If the cells aren't growing well, it's not because the cells can't grow well, it's because the conditions I provided weren't the best. After Irv's advice about how the environment changes the fate of the cells, it led to an experiment that changed my entire life, a whole new experiment, an awareness that actually introduced something brand new to the whole field of biology. And that was the understanding of epigenetics. So the necessary experiment to decide how the environment was controlling all this stems from a simple understanding. I would put one myoblast in a culture dish and it would divide every 10 or 12 hours. First is 1, then there's 2, 4, 8, 16. And I say, after a week, 30,000 cells in the petri dish. Most important fact is, all 30,000 cells came from one parent, so I have 30,000 genetically identical cells. What I do now, here's the experiment. I split the cells into 3 petri dishes, so each petri dish has 10,000 cells, but all 3 dishes have genetically identical cells in them. And what do I do next? I said, I changed the composition of the culture medium because the information was saying that the environment in which the cells were growing controlled the fate of the cells, so I make 3 different versions of culture medium. And so each petri dish gets a different version of culture medium with slight chemistry differences. But remember, all dishes have genetically identical cells. What was the result? In environment A, the cells form muscle in dish 1. But in a different culture medium, environment B, in dish 2, the cells form bone. And in dish 3, again, with genetically identical cells, yet a different culture medium, the cells form fat cells. What's the relevance? That was the clear proof that the fate of the cells were controlled by the conditions of the environment, not by the genetics inherent in the nucleus. Before you blame the cells, as Irv told me, before you blame the cells, consider the conditions of the culture medium. And in the human body, that's blood. Before you blame the cells for pathology, recognize a simple fact that only 1%, or actually less than 1% of illness is connected to genes. Over 90% of illness is connected to how you respond to the environment. And all of a sudden I say, so what's the relevance? Change how you respond to the environment and you change the characteristics of your life. You are the powerful creator, not the victim of your genes.
În următorul videoclip, Bruce Lipton ne dezvăluie care au fost pașii făcuți de 3 cercetători pentru descoperirea spiralei ADN, cum a apărut dogma centrală a biologiei și ce alte descoperiri au schimbat această dogmă. Se credea că totul este fix, stabil, perfect înțeles și cunoscut, dar la doar 10 ani de la descoperirea ADN-ului, o altă descoperire a unei enzime speciale care poate inversa fluxul de informații inițial, și anume ADN, ARN, mesager, proteine, descoperirea enzimei numită transcriptază inversă a demonstrat că fluxul de informații poate fi inversat, astfel că aceasta preia molecula de ARN și o modifică devenind ADN și care în cazul virusului HIV, ea devine o genă mutantă. Enzima revers transcriptază e construită în virus. Aceste descoperiri, împreună cu epigenetica și reacția minții la condițiile de mediu, au demolat formularea inițială a dogmei centrale, deși aceasta încă a mai fost publicată în manualele de biologie până în anul 1990.
In biology, one of the primary principles is referred to as the central dogma. The fundamental simplicity of what the central dogma means is, according to Francis Crick, information in biology only flows in one direction, from the DNA to the RNA and to the protein, which is us. The relevance about this is then our lives are controlled by our DNA. Now I want to say a very important fact because I was teaching the central dogma in medical school to medical doctors. What was I teaching them? Just think about it. That genes control a person's life and a person's life does not affect their genes, and therefore people become victims of their heredity, whatever they inherited that was in their family line. First thing, what is a gene? Well, a gene is a blueprint that is made out of DNA. I say blueprint, but for what? Well, the blueprint is used to create the proteins of the body. And the proteins of the body are the molecules that give us our physical structure and our behavior. And in fact, it's interesting, the term protein means primary, teen, part. So even way back when they identified protein, they knew right away, this is the primary part of a biological system, protein. Proteins are very complex molecules, but proteins also break down very quickly. They're what we say labile, not stabile. Labile means they break down. I go, so why is this relevant? Well, these are very complex molecules that create this whole physical structure, and they break down, we have to replace them. And then the big question came, well, what's responsible for creating the proteins? Well, that was a big intellectual pursuit until 1953. And in 1953, We were provided the insight into the DNA double helix offered by Watson and Crick. Even though, as we've talked about earlier, the real double helix story came from a woman, Rosalind Franklin, and Watson and Crick essentially borrowed her research without her knowing it and came to the conclusions of how DNA is a double helix and the basis of a gene code. DNA is a blueprint, and ultimately this blueprint is going to be used to create protein. But then I have to say, well, it's not a direct blueprint that we use. I say the original blueprints are in the nucleus of the cell. These are the original pieces of information. It's sort of like the Declaration of Independence. We don't use that. We put that away and it's hidden in a vault and nobody messes with it, but we make copies of it. Same thing with DNA. It's very precious. You don't want to mess with it. But what happens is a protein system will make a copy of that DNA blueprint. And that copy of the DNA blueprint is called messenger RNA. And the messenger RNA, the copy, is actually used to create the proteins of the body. So as proteins break down, then what we do is engage a gene to make that protein, engage it by making a copy of that gene called messenger RNA. The messenger RNA goes out into the cell, and then that is used as a template to create the missing protein. So there's a sequence of information and the flow of information. Now we understand it is proteins are the building blocks, RNA are the actual molecules used to make the proteins, but RNA is a copy of the DNA. So we have an understanding of the flow of information that came from Watson and Crick. And it is this, that information in biology only flows in one direction. It starts with the DNA, goes to the messenger RNA, which is a copy of the DNA, and then that is used as a template to make the protein. You, as the proteins that are derived from this central dogma, cannot go back and alter the original genes. This is the belief that led us to understand that we are victims of our heredity. Whatever genes you have, they control your life, and you do not control the genes. This is the story of the central dogma, a unilateral flow, DNA, RNA, protein. Very interestingly, as a professor in a medical school, I was teaching medical students the concept of the central dogma. It was only after I left the medical school that I actually looked up the meaning of the word dogma. And the significance blew my mind because dogma means a belief based on religious persuasion and not scientific fact. And all of a sudden I started to realize I have been teaching religion in medical school when I have been teaching what is called the central dogma. Well, how did Francis Crick come up with the word dogma? And the answer is, when it was actually confronted with the truth of what dogma meant, he acknowledged, I didn't really understand the word, and I used it, but a much better word would have been hypothesis, which is more accurate. So instead of the central dogma, what we're really referring to is the central hypothesis that information goes DNA, RNA, and protein. Well, a hypothesis, by definition, is not a scientific fact, it's a suggestion. In 1958, when Francis Crick offered the central dogma to the scientific community, they just thought this was the greatest of revelations, something like Moses coming with the 10 Commandments of how biology works. The fact that we go DNA, RNA, and protein, they were looking for an answer like that, and when it came out, they said, it sounds great. They accepted it. But what was the point? The point was, it was never scientifically assessed. It was a hypothesis from day one, but it was repeated in textbooks from 59, 69, 79, 89, 99, still in the textbooks, that the central dogma is an accurate scientific understanding of the flow of information in biology. No, it's not. It was a hypothesis and never proven to be true, just repeated so frequently that people accepted it as truth. Everything changed in 1969 when Howard Temin, working at the University of Wisconsin, working on the HIV virus, found something totally unique and actually was chastised for his findings. What did he find? He found that the HIV virus is an RNA virus. But what's unique about it? There was also an enzyme attached to that virus called reverse transcriptase. And what was that? Well, Howard Temin found out that this enzyme took the RNA molecule and reverse programmed it back into the DNA, so that the HIV starts out as an RNA, but ultimately becomes a gene in the cells that are infected with this virus, reversing the flow of information. Well, you have to have seen what happened to Howard Temin when he was trying to present this to the public, because all the central dogma people jumped on him and said, no, information only flows in one direction. But between Temin's research and others, such as David Baltimore, another famous virologist, they all came to the same conclusion. Reverse transcriptase is an enzyme that will take messenger RNA and alter the DNA, going back against the unilateral flow. And all of a sudden it says, well, first thing, oh my goodness, the central dogma is wrong. RNA can actually go back into DNA. In his HIV study, the reverse transcriptase enzyme is actually built into the virus. That would suggest that, oh no, regular messenger RNA cannot go back into the DNA without this very special enzyme, reverse transcriptase. But guess what? Once the human genome was completed, it was recognized that 10% of our DNA is actually coming from viruses and virus-associated proteins. It is now recognized, built into our own DNA, we already have reverse transcriptase. Well, the predominant use of that reverse transcriptase deals with something called telomeres, extensions of the DNA. That's not the only reverse transcriptase in our nucleus of our cells. The other reverse transcriptase can do exactly what the same enzyme did with the HIV virus. We have built into our genes reverse transcriptase molecules that can take messenger RNA and put it back into the DNA. And so all of a sudden, it really says that reverse transcriptase is not just isolated in viruses. We each have our own reverse transcriptase molecules in all of our cells because it's part of our genome. It was such an important finding that Harold Temin got a Nobel Prize for identifying reverse transcriptase, an enzyme that will take RNA and put it back into the DNA as a gene. Well, that was the start. But by 1990, a bigger explosion in central dogma occurred, and that is the insight that led to a new science called epigenetics. Epigenetics is the science how environment alters the readout of the gene. I go, how can it do that? Because the nature of the system is nothing goes back and alters the gene. Well, Temin's reverse transcriptase challenged that for sure. But what did they find out? That the chromosome, the hereditary units in our cells, are actually 50% DNA and 50% protein. I go, so what? I said, well, in studying the DNA story, scientists routinely separated the protein from the DNA of the chromosome, threw away the protein, and just worked on pure DNA. What did they get from this story? Well, they found out, yes, the DNA programs a protein, which they always thought, but the big question was then what's programming the DNA? And they did this experiment. And I said, what was it? They took a DNA double helix, they separated one of the helices, so now it's a single helix, and they take that single helix, put it into a beaker with the building blocks of DNA, and incubate it. But when they pulled out what was once a single DNA molecule, guess what? When they pulled it out, it was actually turned back into a DNA double helix. The DNA coded for its own complement. In other words, DNA codes for DNA. And all of a sudden, the answer came very clear. DNA makes proteins, DNA makes itself, central dogma. DNA is in charge. Well, problem? What about the 50% of the proteins they threw out? What were they doing? 50% of the chromosome is proteins. They said, that's not relevant. We just want to study DNA. So I said, why is this a problem? First thing is this, there's no place in a biological cell where there's something called free DNA double helix. Every time you see DNA in a cell, it's always complex with proteins. So the idea I can study DNA by itself and remove the proteins is an artifact. Why? There's no place in biology where DNA exists by itself. And so the study that showed that DNA can reproduce itself, artifact, totally wrong. Why? It's not natural biology. So I said, what did we find out in 1990? And this is actually, I was doing the pioneering research on this in 1967, but it took until 1990 to recognize epigenetics. And I said, what is it? It's how the environment alters the proteins on the chromosome, but the proteins control the activity of the DNA. While the original story of the central dogma concluded with what we refer to as the primacy of DNA in controlling your life, meaning your life is an expression of your DNA and you have no power to control that. That was the hypothesis. Well, that hypothesis is now incorrect, because now when we put all the information together, how the information flows through the system, it doesn't start with DNA, RNA, and protein. It starts even higher than that. It starts with environmental signal affecting chromosomal proteins, which in turn affects the DNA readout, which affects the RNA readout, which ultimately affects the body in itself. So the whole idea is not the primacy of DNA anymore. If you look at the flow of information, the top level of that flow is the primacy of the environment. And this basically says how you respond to the world in which you live. The environment you're responding to is actually controlling your genetic activity. And I say, and the relevance about that is, well, central dogma made you a victim of your heredity. You can't control the DNA. That's false. We now recognize that the environment, and more specifically, our perception of that environment, actually is what leads off the control of the DNA reading to get you to the protein. And all of a sudden, we go from victim, primacy of DNA, to master, primacy of the environment, because I can change that environment, I can change my perception of the environment, and in doing so, I change the readout of my genes. I'm not a victim. I'm a master of my heredity. But the most important fact is this: we must recognize that those that are still touting the power of the central dogma, this is a very false statement. The idea is, no, DNA is not the control of your life. As we talk about in the biology belief, consciousness, your perception, your interpretation of the world is what controls your genes. Since you can change that, that means then you can change the genetic readout, which then means you are not a victim of your DNA, you are the master of your DNA.
În încheiere mai prezentăm un material video realizat de Bruce Lipton cu privire la exosomi. Ce sunt aceste particule, ce rol au și cu ce ne ajută anumite teste sanguine care evidențiază efectele acestora? Explicațiile profesorului Lipton sunt absolut fascinante și merită să le ascultați cu atenție.
There's a new story we've touched upon in the past, a story about something called exosomes. Exosomes are little pieces of cells that pinch off as little tiny vesicles. So small you can't see them in a regular microscope actually easily. And these exosomes were thought to be degradation products. In other words, a cell was removing a piece of the membrane and cytoplasm, and so that it would be broken down and the building blocks recycled. So exosomes originally thought were just a way to remove parts of the cell that need to be replaced. But as I started to notice, many of these exosomes were not degradation particles, but they actually contained information, DNA, RNA, proteins that would affect a cell if that exosome was picked up by another cell. So cell A releases an exosome with information that controls the genetics of a target cell. Most important in regard to cancer that we're just talking about, a cancer cell can arise anywhere in the body. That's a fact. Could arise anywhere. But there are very preferred sites where cancer grows, such as the liver and the lungs, pancreas, different sites where cancer seemed to be manifesting. And our original thought was, oh, the cancer was created in that organ. Oh, the cancer formed in the lung, or the cancer formed in the liver. Well, there's a whole new story. Exosomes are vesicles that pinch off a cell containing molecules that control the behavior of a cell that will pick it up, controlling the genetics and the protein synthesis and all that. So by definition, an exosome is a virus. It carries a piece of information from cell A to cell B. And cell B isn't random, meaning on the surface of the exosome, which is a membrane-wrapped structure with some cytoplasm inside containing nucleic acids and control factors, such as things that control the immune system, that these exosomes are information carrying structures that are picked up by a cell. The cell that picks it up, again, it's not an accident because on the surface of those exosomes are receptors that complement specific cell types. So let's say a precancerous cell is forming in the leg. I go, then what? Well, we now know that those cells release exosomes. I go, yeah, but this information carrying vesicle, what does it have? Well, first of all, it's directed to a site, the liver, the lung, the pancreas. So that the cell releases these information containing virus particles, I'm going to just give it a name, virus, that is sent to a specific site, and the cells at that site pick up these viruses, and those cells then change their function based on what information was in that exosome. And all of a sudden I said, oh my God, it's called terraforming. In other words, the precancer cell releases the exosomes. The exosomes go to the site where the cancer will grow, which is not where the precursor cell is in the first place. As I said, precursor cell could be in the leg, but the exosome is directed to cells in the liver. When the exosome gets to the liver and the cells pick it up, they change their function to create an environment, terraforming, that will support the cancer cell, which is going to travel there in the future. Oh my God, a cell in some place in the body sends out a signal to a destination, such as liver or lung, and says, prepare the site because this cell is going to move from where it is now. It's going to go through the system and end up in the liver or the lung or that destination where the exosomes were. So that the exosomes carry information to another part of the body to prepare for a cancer. And all of a sudden I go, wow, then these exosomes are not just degradation things. No, these are information containing viruses that come from a cell source, but have a very specific destination. It's not accident or chance. It's like, oh my God, we never understood anything like this. We never knew our own cells are communicating at another level because we think in the body, communication is nervous system is communicating, but nervous system only actually contacts a very small percentage of the 50 trillion cells in your body. So not all cells are directly connected to the nervous system. Another way of communicating with the 50 trillion cells is to release hormones or, neurofactors that go in the blood. And I go, well, that's really interesting because that is a way of coordinating the body because the blood is going to travel through the body carrying this information, and the cells that are responding to the information are going to adjust their behavior based on what the hormones are. Yet there's a problem. And the problem is this. There are two very important messages that cannot be trusted to the nervous system because not enough cells are connected to it. And it can surely not be connected to hormones because all cells are going to be exposed to it. And I said, these two messages are the message to reproduce more or the message to die. If those messages go to the wrong place, the whole system could collapse. So I said, well, how do you assure that a message to reproduce is going to go to the right place or a message to die is going to go to the right cell? And the answer is, exosomes on the surface have these receptors that determine the destination and which cell is going to pick it up. So sending exosomes is one of the highest levels of communication. It's like a zip-coded telegram that is being sent. Now back up. Exosomes are in the blood. They're all over the place. How many exosomes, if I took a blood sample from every one of you right now, I said, let's count the exosomes in your blood. Here's how many are going to be in there for the average person. 2,000 trillion. Okay, that's a big number. Okay, let's put it in another word. 2 quadrillion. Well, millions, billions, trillions, quadrillions. That's a number that's mind-boggling. Right now, every one of us has 2 quadrillion exosomes in our body. But guess what? When a person has cancer, they have 4 quadrillion exosomes in the blood. What's the point? You can see a person is going to develop cancer by counting the population of exosomes. If they go above the 2 quadrillion and start going to 3 or 4 quadrillion, that's an internal message system that says a cancer is evolving. So guess what? Now we can even do a survey of how many of these exosomes are present in a body, which is indicative of a future cancer unfolding. So, oh my God, there's a test that can let you know if you're moving toward cancer. So the whole idea about that is simply this. We go back to the family with the gastro problem where the stomach creates a cancer. I say, before you do anything radical like cut the stomach out, there are 2 things that we should be doing. Number 1, assessing what is the common psychology of those in the family that get the stomach cancer versus those 25% that don't get the stomach cancer, because there's a psychological input to this. It's not an accident. Cancer is not an accident. It is well choreographed, well planned. So we can start to say, well, what psychology is behind those that end up having the cancer? And number 2, before you do anything radical, you can do a survey of the exosomes. And therefore, before you cut somebody's stomach out, you can see, are they moving toward a cancer or not by looking at that population. I bring up exosomes because this is brand new in the sense of meaning of what's relevant to our lives, to recognize that our own cells create viruses. Now, the problem with the word virus, as soon as you say the word virus, a lot of people get like a stomach tightens up and go, horrible, viruses are scary, viruses cause all the problems. And it's important to note, very important, biology doesn't create something that's all bad. Well, we focus on viruses being bad. We might have to ask, is there a good point to viruses? And the answer is absolutely. Because viruses are communications that are so specific, they can be sent to specific cells out of 50 trillion. You can target a very specific cell type if you have a virus with a zip code receptor system that only identifies that cell type. What's the point? It is the most secure form of communication in a biological system, exosomes. They're new. Their understanding is just coming into the world. I need you to understand this because let's stay ahead of the curve on things. Exosomes are found. Exosomes coordinate the community of 50 trillion cells so they communicate with each other and create an environment that supports what's going on. Now, if it's a negative environment, then of course, then we have to deal with things like cancer and pathology. But the same viruses are very important for normal functions. I'll give you one example where this virus is so important, exosome virus, and that is this. When a woman is pregnant, recognize that the placenta and the fetus have different genetics than the mother because they represent the composite genes of the father and the mother together. So fetal cells don't have the same genetics as the mother. And I say, well, why is it relevant? Because the placenta, the cells of the placenta merge with the uterus and penetrate the uterus. Finger-like projections of placenta go deep into the uterus where they contact the mother's blood supply and open up a passageway so blood can go from the mother system into the fetus and nourish the fetus and help the development of the fetus. I go, well, wait a minute. By all rules, the mother should not be able to get pregnant because if the foreign cells start to invade her uterus, that's the equivalent of cancer. And by definition, the mother's immune system should reject the fetus. I go, well, obviously it doesn't. How can the mother's immune system be prevented from rejecting the foreign fetus? And here's what we now know. Exosomes. The placental cells make exosomes, particles in membranes with control information that control the immune system of the mother. Inside are special molecules that are actually hormones for the immune system. They're called cytokines. Cytokines manipulate the function of the immune system. Well, the foreign fetal cells make exosomes, virus-like particles that are picked up by the mother's immune system. And these virus-like particles redirect the immune system to go away from the placenta so that the immune system is not recognizing the foreign fetal tissue, because the foreign fetal tissue sends viruses with cytokines that shunt the function of the immune system and push it to go somewhere away so that the placenta can grow and mix its cells with the mother's cells. It's even been suggested that at term, when the baby is being born, part of it is an immune rejection, that birth is an immune rejection, that the mother's system at birth pushes the placenta away, disconnects it from the mother's uterus, and in that process engages in birth. So I wanted to introduce the exosomes because I talked about them before, but they're becoming more and more a topic of discussion. And so you won't be unaware of the future of what's happening in this biology once we start to recognize that our own cells are communicating with each other through viruses.
Acestea au fost doar câteva din numeroasele videoclipuri produse și prezentate de profesorul de biologie Dr. Bruce Lipton și sper că au conturat câteva idei despre modul în care ar fi bine să abordăm viața, biologia, credințele și realitatea de care avem nevoie. Fiecare avem propriile repere de viață, propriile valori față de care ne raportăm și pe care le urmăm. Avem propriile credințe și toate acestea la un loc conturează modul în care viața noastră se derulează. Dacă lucrurile nu merg așa cum ne-am dorit, atunci avem nevoie de schimbări în stilul de viață. Cu cât mai grave sunt aceste probleme, cu atât mai intense, profunde și dificile vor fi aceste schimbări. Așa că avem nevoie de exemple vii care să vină din partea celor care au reușit. Iar Bruce Lipton este cel mai bun exemplu pe care vi l-am putut oferi în nenumărate episoade difuzate până în prezent. Vă mulțumesc pentru faptul că vizionați această emisiune în fiecare săptămână la TVR Cluj sau online pe internet și vă invit data viitoare la o nouă întâlnire cu emisiunea dumneavoastră preferată, Știință și Cunoaștere.